Hi, welcome to this lecture on EnvAdapt adaptation of animals. Now we will look more closer on the mammalian kidneys, so how we can regulate the water control. So over here is the overview of the nephron, and now it's not only having these tubules that were present in the amphibians, but now also this long tube. And why we have the long tube there is very important. So we have the primary urine with water, with urea. So we can get rid of the water, we can get rid of the urea, but at the same time, there's a plenty of good stuff. There is glucose, there is amino acids that we definitely don't want to lose, and therefore we want to take them back. And where we want to take it back immediately. We use the proximal tubule in the same way as in the amphibians. So we are taking sodium, we are taking chloride, and the most important, we are taking the nutrients back. But as you can see, it's also that we are at the same time we are pushing a little bit ammonia, taking back carbonate, taking back water. But this water take back, because it's working in the same way as in the amphibians, it's not very effective. So still the osmotic pressure is quite low. And then we are getting the urine on this descending limb of Henle, where we are taking away water. Water leaves passively. There is no water pump on this planet at all. And then we have the thin ascending limb and the thick ascending limb, and in both cases we are taking away sodium chloride. And the idea is that, okay, when you are taking more sodium and chloride from this urine, you are increasing their concentration in this interstitial fluid. And then when the urine is going up, it's there in the distal tubule, and in the distal tubule we are pushing some potassium there, pushing some protons, but taking still carbonate and sodium chloride and some water away from the urine. And then the third round through this medulla is on the collective tubules. We are actively taking again sodium and chloride away. That is then affecting on the higher concentration of sodium and chloride in the medulla, and then we are taking away passively water and on urine, that we are going to discuss in the end of this video. Okay, why these tubes are going up and down, down and up and down? So they are going side by side. And so we have descending limb and ascending limb. They are almost touching each other. There is only a small gap between them as the interstitial fluid. So therefore, ions that we have here, ions that are in this interstitial fluid, and the ions in this descending limb, these ion concentrations are interacting, as well as how much water we have in the ascending limb, in the interstitial fluid, in the descending limb. So what is the osmotic pressure? So when we are moving these ions from here to interstitial fluid, these ions can be moving already also in this descending limb. And this is the basic mechanism, a little bit old school stuff, that okay, in this ascending limb we have the pump. We are using ATP to pump sodium and chloride away from this urine. And they are of course not going with the same protein, but anyway, the idea is that okay, we are using active transportation, and then we are raising this concentration, and we are raising the osmotic pressure. And in that case, when we have high concentration, then sodium wants to go in, chloride wants to go in on this tubule, and if there is a pathway, then of course they can go. And if there is a pathway for water, then this water will live to be in the isoosmotic conditions with this environment. And therefore we have the single effect. So in this tube that is going up, we are moving with ATP to moving the sodium and chloride. So therefore we have higher concentration over here. And therefore we can move these ions over there, so the concentration is rising. But at the same time the osmotic pressure was rising or higher over there, so water is moving away from this descending limb. And it works in even more interesting in the Henle loop, because there's all the time flow of this urine. So the active transportation, so we are moving sodium and chloride. That causes that, okay, the osmotic pressure is increased, and therefore the osmotic pressure is increasing also in this descending rib, because we have ions moving in this tube, and the water is moving out from it. And therefore in this downward going flow solution, the pressure, osmotic pressure, is higher than in this tube where the solution is going up. But as we get more and more primary urine, so this is moving downward. So therefore we are having end-to-end gradient. So over here in the lower part, the gradient is much higher than in the surface. Okay, how it works? Let's assume that, okay, the kidneys in the beginning, it's isoosmotic. So everywhere we have 300 milliosm pressure. But when we are using the ATP to pump sodium and chloride, then the sodium and chloride are moving on this descending limb, and water is moving out from it. So that increases the osmotic pressure in this around the tube, but also in this tube that is going down. So that was the single effect. But now the solution is moving, and that will rise the pressure also in this ascending limb, for which now, when there's again active transportation, now the interstitial fluid will be having even higher osmotic pressure, for which we have even higher osmotic pressure in this descending limb. And then, of course, again the solution is moving. And therefore we have even higher concentrations and osmotic pressure there in the rising tube, which from which again sodium and chloride will be pumped, and therefore we are increasing and increasing this osmotic pressure there in the interstitial fluid, as also in the end of this descending limb. Okay, this is a little bit too simplified, because the active transportation happens only in the upper part. So in this lower part, it's actually going with by different ion channels, the sodium and chloride out. But we will discuss it a little bit later. It's a little bit complex way. So, but it's still, although it's old school and not very accurate, it still works. So over here we can see that this countercurrent is multiplication is increasing this osmotic pressure, but to about 1000 milliosm. So over here we have 1000, over here we have 200. So it will increase not in the outer zone but in the inner zone very drastically. And these cells must be surviving this hyperosmotic environment. And therefore, they are not doing it the way that, okay, inside the cell there would be 200 milliosm and outside 1000, because otherwise the water would just leave the cells. No, that is not the case. And notice that neither is the case that, okay, you are increasing sodium concentration. No, they are not, can be any more regulated. The idea is that you are increasing something that is not lethal. You can have polyols, like sorbitol, or these trimethylamines. So these trimethylamines we were learning from the shark, with high concentration of urea. So something that is not toxic, you can have high concentration. And these polyols, from where these are familiar for us, it was on the inhibition of getting frozen. So we have the freeze avoidance. But of course, the freeze avoidance doesn't have anything to do with the human kidneys, but we can use the same product. And the idea is that, okay, we are using something that is non-toxic. And in the intermediate, the concentrations can be very much higher than in the cortex. Okay, how this water balance is then regulated? Well, when we are getting in this descending limb, we can see that this osmotic pressure is getting higher because we are losing water. And getting more sodium and chloride there. But then, why it's not losing the water on the ascending limb is because this is water impermeable. There is no aquaporin, so therefore this solution, the water is not going away. But because we are getting less and taking all the ions away, so actually it's quite diluted in this distal convoluted tubules. So the osmotic pressure is lower than in the blood. And then when we are taking the water away in this collective duct, then this getting again highly concentrated. So because it wants to have the same osmotic pressure as the surrounding. So in that case, it just depends on how much sodium and chloride we are pushing, or whatever substance in this in the outside of these tubules. So this is affecting how concentrated urine we can secrete. But then, if we have a diuresis, so when we want to get rid of the water, the idea is that, okay, now even these are water impermeable. So there is no pathway to get the water out. So therefore we can have very diluted urine. And how we can modulate this water permeability is by using the aquaporins. So when we are using the antidiuretic hormone, we are increasing the number of aquaporins on the membrane by 3 times. But at the same time, we are increasing the permeability to 10-fold. So it's a huge difference in taking the water away from this urine. Okay, now we've been discussing that, okay, we are taking actively sodium and chloride or whatever. So let's look a little bit more closer, because when we are in the proximal tubule, we have in this primary urine, we have high chloride, we are having quite low carbonate concentrations. And in the epithelial, we keep the sodium concentration quite low, and the voltage negative. So therefore, keep the negative voltage, we are allowing potassium to flow from the epithelial cell, and then pumping the sodium away with the sodium-potassium ATPase. And for that reason, we have a very low concentration of sodium, so the sodium is wanting to go inside the cell. And with that, we can get all kind of molecules. And from the amino acids that we are taking, they are used for energy production in these epithelial cells. And therefore, we are producing ammonia as a byproduct that we can get rid by exchanging it to sodium. And one thing that is also done is, okay, we are changing the protons to sodium, and these protons are needed that, okay, when we have carbonate, it will interact with the proton to produce carbon dioxide. And now this carbon dioxide is permeable through these membranes, and then we can get that way we can get this carbonate away from the urine. And then on the thick ascending limb, where we are pumping sodium and chloride by using the same sodium-potassium ATPase. So actually, when we are taking the sodium and chloride away from the urine, we are not directly using the energy. We are using it by the sodium-potassium ATPase. And therefore we use a sodium-potassium chloride co-transporter. I think we were discussing this earlier, as well as a sodium chloride co-transporter. And then in this, when we are getting third time to these deeper parts on these collective tissues, we have three different cell types. We have collecting duct B1C cells, where we are forming carbonate. And this carbonate can be used to get chloride away from it. Then we have these principal cells that are used to take the sodium away. And surprise, surprise, by using the sodium potassium ATPase. And with these mechanisms, we can then affect in these principal cells that, okay, we can have also water channels to take also the water away from it. And then we have these third cells that are using pretty much the same way that, okay, producing carbonate and then changing the chloride concentrations, etc. So that's why we can also reduce get rid of a little bit ammonia. Okay, but so now we have learned that, okay, on which mechanisms we are putting hydrogen or ammonia there in the tube. They are because we are producing using for these nutrients for energy metabolism, and then by how we are using taking the sodium chloride away or on different places. Usually it's because we have sodium potassium ATPase. And one thing that we have not yet discussed is that, okay, over here we have the urea. So in this, the late part of this collective tissue tubule is not permeable for any of these substances. Instead, only on water and the urea. And the idea is that, okay, when you are getting the urea, you can have very high concentration of urea there. And therefore you can have quite high osmotic pressure, but not having a lot of sodium or not having a lot of chloride there. And therefore the sodium and chloride can still go out passively. Thank you.